After biopsy, the isolated tissue is fixed in the Bowen's fluid. It is called as a compound fixative because it is made up of picric acid, formalin, and acetic acid. Bowen's solution is used to avoid shrinking of tissue. Tissue is removed from Bowen's solution after 24 hours of fixation and afterwards processed in increasing grades of ethanol, each for 15 minutes. Further, the clearing of tissue is carried out with the help of xylene. In next step, impregnation is carried out using paraffin wax having melting point 60 degrees Celsius. Before the direct use of wax, it is filtered twice by using filter paper to remove impurities. In impregnation, xylene is replaced with molten paraffin wax. Tissue is kept in xylon made up of 1:1 wax and xylene for 10 minutes. Later, 3 changes of wax are given with interval of 10 minutes. By using Leuchart's L pieces, the tissue is oriented in molten wax, which is called as embedding of the tissue. These L pieces are arranged according to size of the tissue. The glycerol is applied on the L pieces and the bottom surface to facilitate easy separation of wax block after solidification. The molten paraffin wax is poured between L pieces and allowed to solidify at the bottom. To avoid solidification of surface layer, a hot dissecting spatula is slowly moved through the upper layer of wax. As to get vertical section or transverse section, the tissue is placed in the wax accordingly and left to solidify. Plastic blocks are also used for the casting. After 30 minutes, the block is trimmed to remove excess wax so the tissue could be seen from outside. The trimmed block is then fixed on the block holder. The front side of the block holder is covered with a thick layer of wax in which the block is fixed by melting the central part of wax and cementing with the molten wax is carried out for tightening of the block. Microdome is used to get thin sections of the tissue embedded in the paraffin wax. In order to move the wheel, the safety knob must be turned downward and handle must be pulled outwards. The handle on the right side of the microdome is used to move block across the blade. The block is later fixed in the block holder and tightened with the help of adjustment knob. Once the block is ready for sectioning, remove knife guard and adjust the position of the blade. Set thickness of the section as per requirement and start sectioning. For getting perfect ribbon of the tissue, the sectioning must be done at a constant speed. Sections get attached one after another to form a ribbon. After sectioning, a long ribbon is obtained which could be cut into small pieces with the help of scissors according to the size of slide on which ribbon is to spread. Meyer's albumin is used as an adhesive which consists of glycerol, egg albumin and thymol crystals. It is smeared on the slide so that the sections stick well to the slides. Then slide is flooded with the distilled water and the sections are placed on the slide with the help of brush and forceps. Afterwards, slides are kept on the heating mantle for a few minutes to remove wrinkles on the wax ribbon. After spreading, excess water is removed from the slide and slide is kept for drying. Staining is required to visualize the tissue structure. The slides are first kept in xylene for complete removal of wax and then are passed through rehydration process because hematoxylin is a water-based stain. After staining with hematoxylin, slides are passed through dehydration process because eosin is prepared in alcohol. Few drops of DPX are spilled over the slide for permanent mounting. While putting the cover glass, air bubbles are avoided. Slides are kept for drying overnight.
You started with biopsy by taking a tissue, and now you are observing the stained tissue, how it is organized at tissue level and interestingly at cell level, how its nucleus, cytoplasm and cell organelles are seen. It is very interesting to understand the functioning of the entire organ system. Friends, such a beautiful techniques is very widely used in medical field to diagnose the diseases, and in research it has a great applications with reference to some studies of toxicity or even any abnormalities changes occur into the body and the organ system. This science, these techniques will open a new window of knowledge to the student to understand the organ system and its function. Thank you.